Hello everyone, this is the second video from my YouTube channel. Today we are going to discuss about a topic from class 12th standard, that is sexual reproduction in flowering plants, and this will be the first part of the topic wherein we are going to discuss about the microsporogenesis and microgametogenesis. So just to start with the chapter, or to introduce the chapter with you, we will be discussing a little bit portion from our class 11 morphology part, wherein we have discussed about a complete flower. We have discussed about a complete flower in class 11, and while I'm talking about the complete flower, we know that a complete flower, it has two different structures, or rather the four holes of a complete flower can be classified into two. One which is the essential part of the flower, and the other one is the non-essential part of the flower. One is the essential part of the flower, the other one is the non-essential part of the flower. So in the non-essential part, we generally include the sepals and petals. That means the outermost hole, which we call it as calyx, the individual units are known as the sepals, and the innermost hole, the second hole, which is the corolla, individual units are known as the petals. Whereas the next two inner holes, that is the androecium, which is the male segment or the male reproductive part, and the gynoecium, which is the female reproductive part. The androecium do contain two different parts, that is the anther and the filament. Though the anther and the filament, the individual unit is known as stamen. The anther and the filament of the stamen, it is connected with a small structure, a stretch of tissue known as the connective. And the gynoecium consists of the carpel, the pistils, and this pistil, it comprises of the stigma, the style, and the ovary. So the entire event of the sexual reproduction in the flowering plant, in a flowering plants we call it as, generally we can call it as angiospermic plants also, the one which forms flower, so or produces flower. Now in this segment we are going to discuss about the different events which are related to the sexual reproduction in plants. The events which takes place in a sexually reproducing plant where they form flowers, it comprises of three events, that is pre-fertilization events, fertilization events, and the post-fertilization events. Today we are going to discuss about the first part, that is the pre-fertilization events. The pre-fertilization events, it comprises of the formation of the gametes, the male gametes and the egg, wherein we will discuss about the first topic we will be going for, that is the pollen grain formation or the microsporogenesis. So we will discuss about the microsporogenesis. To discuss about the microsporogenesis, sporogenesis means formation of the microspore. So here we will be discussing about the pollen grain first. So just So just to discuss about the structure of the stamen, I'll just draw a simple diagram of the stamen, which we already have done in class 11 level. That is, these are the bithecous or dithecous anthers or bilobed anthers, and this is the filament. So if we go for a transverse section through this, if we section or chop it like this, we will be able to see some structure like this. Wherein on the four corners we'll see some sac-like structures. These sac-like structures are known as pollen sacs, wherein innumerable number of pollen grains are formed. Which is the male reproductive unit of a flower. This structure forms the stromium. Now, we will be discussing this portion in detail. We will be discussing about the development of the pollen grain or the microsporogenesis. Now, before we go for that, I'll just draw one portion of this, how it looks under the microscope. So, the portion do have some levels. I'm just drawing one half of it, an arc of it. It has an outer epidermal region, which we call it as the epidermis, followed by the endodermis, then there is nearly 2 to 4 layer thick structure which is known as the middle layer. These are the middle layers, followed by a nutritive layer known as the tapetum. This tapetum provides nutrition to the growing pollen grains or developing pollen grains. And inert to this are the pollen grains. So these are the microspore mother cells which forms the pollen grains. We call them as MMCs, or this can be called as a microspore mother cell, or they are known as the pollen mother cells. So we'll discuss about the development of them. So we are going to discuss about the development of the pollen grains, wherein the hypodermal cells of the anther, which upon differentiation forms the archisporial cells. The archisporial cell have two characteristics with very interesting characteristic feature apart from the hypodermal cell, that is they have a prominent nucleus and they have abundant cytoplasm, or you can say abundant protoplasm as well. So this undergoes a periclinal division like this, oblique division, okay, horizontal, longitudinal. So they undergoes a periclinal division, as in like this. This is known as periclinal division, P, and this is known as anticlinal division. So they undergo a periclinal division and they form and the inner layer from the sporogenous cells. This forms the inner layer. Now, of course, the outer layer, it undergoes this anticlinal division. And it forms nearly the 2 to 5 layers of anther wall and tapetum. And the inner wall, the inner segment, inner division, the sporogenous cells which were formed, they undergo a specialization, a special characterization rather, or differentiation, and they form the microspore mother cells, which we can abbreviate them as MMCs, or pollen mother cells. PMCs. This microspore mother cell or the pollen mother cell, they undergo meiotic division. And they form the microspore or the pollen grain, which forms a tetrad. A tetrad means they form a tetrad. A tetrad as in, it can be like this. That is, they are forming a tetrahedral structure. Or tetrad means the four of them, they undergo the karyokinesis, but the cytokinesis, the final cytokinesis doesn't occur in the telophase 2. So they are still in the telophase 2 stage. So they are not completely separated from them. So either it can be tetrahedral or it can be isobilateral like this. Or they can be linear, but the most common one is the tetrahedral. So this microspore undergo, after they go through their dehydration, they becomes individual cell, and they become the individual cell and they form the separated pollen grain, the matured one. Now this separated pollen grain, they undergo maturation. And finally they form the matured pollen grain. Now this separated pollen grain and the matured pollen grain, what is their basic difference? This I'll speak about them. Now the pollen grain, after their separation, they form a structure like this, wherein they have an inner intine and an outer exine. This exine is comprised of a chemical which is the strongest chemical. We can see the chemical substance is known as sporopollenin. This sporopollenin is the most resistant biological substance known. Fine. Whereas this is made up of, the exine is made up of sporopollenin, the most resistant biological material till known. And the intine is very basic. It is made up of cellulose and pectin material. Now inside this we have a vegetative nucleus and a generative nucleus. Which later act as a vegetative cell and a generative cell. This this vegetative cell and generative cell, this when undergoes the maturation, they form a three-celled stage structure, which is the matured pollen grain. They have a germ pore through which the pollen tube will grow, and they have the generative cell. The generative cell, it forms the male gamete, and the vegetative cell forms the tube nucleus. So this is in this stage. So these are the male gametes, and this is the tube nucleus. So hereby the pollen grain is formed. So till here we will discuss in this video. In the next video we will be discussing about the embryo sac formation, and subsequently we will be discussing about the Microsporogenesis. Thank you. Till then, stay tuned and do follow and subscribe my channel. Thank you so much.